So I hope you all enjoyed that wonderful lunch. And again, that was sponsored by Corbion, so be sure to thank Jorge if you see him. We're going to start the afternoon off with a panel on digitizing biology and biomanufacturing, and it is a star-studded panel, as all of them. John Cumbers from SynBioBeta will moderate the next panel that focuses on the advent of synthetic biology and artificial intelligence as it applies to biomanufacturing. And with that, I'll turn it over to you.
Thank you, Blake. Can everybody hear me okay? Not yet. Can everybody hear me okay now? Let me keep talking like this and see whether you can hear me now. I can hear my own voice now. Okay, wonderful. Well, I just want to say a big round of applause for Blake and Mary and the rest of the crew here for putting on an amazing event in a wonderful venue. And as a professional meeting organizer, as I am, I want to also commend the long lunches, because it's actually the opportunity that we all have to interact and speak with each other that makes these events so special. And what a high caliber of people you've brought together today and tomorrow for this event. So thank you. My name's John Cumbers, and I'm the founder and the CEO of SynBioBeta. And SynBioBeta is an activity hub for the synthetic biology industry. So we bring together startups, investors, larger companies, policymakers all together for a big showdown once a year here in the Bay Area, with various events happening throughout the year and the rest of the world throughout. And we also produce a weekly newsletter. So if you're interested in finding out what's hot and what's not in the industry, then you can sign up to our newsletter. I was chatting with Mary over the lunch break, and I said something, and she said, well, I want you to start your panel with that to explain to people about it. So what I said to her was that on my tombstone are going to be written the words, he built the Taj Mahal out of mushrooms in paradise. I will allude to what that's about at the end of the panel to keep you guessing. But I promised Mary that I would start the panel with that for some light entertainment. So the panel is all about the impact of digital technologies on biology, and we have an amazing group of panelists to discuss that topic. I'm going to go across the panelists, and each panelist is going to give a short introduction about who they are and what they do. And then we're going to talk a little bit about some of the digital technologies that are currently impacting biology. And we're going to look at what's changed over the last 10 years in the engineering of biology. And then we're going to throw it open to the audience, as I know we've got some great questions about these kinds of topics that we're going to get into. So without further ado, Darlene Solomon, the CTO of Agilent. Welcome, Darlene.
Great. Thank you, John, and great to see everybody today. Lots of old friends and many new ones being here. In terms of who I am, John covered that. So maybe I'll just say a little bit about Agilent for those who maybe are less familiar. So we're a global leader in life sciences, diagnostics, and the applied chemical markets. So applied chemical is food, environmental, chemical, energy, major industrial. And in general, servicing the future transformation of the analytical laboratory. We're a leading provider today, I think something like 265,000 customers worldwide in chromatography and mass spec and spectroscopy, many of the technologies that help understand metabolomics, glycomics, lipidomics, protein characterization, things like that. The analytical lab these days, the instrumentation is important, but it's kind of an incidental piece compared to where the opportunities are in the front end sample prep and back end getting actionable information out of all that data. The diagnostics side is primarily pathology and cancer diagnostics. We work very closely with pharma in areas like companion diagnostics. I'm sure everybody here has been touched directly or indirectly through the amazing advancements in immuno-oncology these days. And so like PD-L1, which is the companion diagnostics for the first drugs out of Merck and BMS, the diagnostic for that is all Agilent. And just kind of more looking forward, cell engineering, cell analysis, live cell analysis, biopharma are all major areas for us of interest. And maybe kind of in the spirit of this meeting, we're not necessarily producing an end product. We're really more enabling research, enabling manufacturing. But sustainability is a really important part of the company. And that's in how we do our own internal operations. It's with an eye to making our products more sustainable, rather, that can be in, for example, not using, rare gases, not using pump oil inside of pumps, being able to eliminate those types of chemical usage, as well as reducing the footprint and power consumption and exhaust, things like that from instruments as well. And in fact, we've gotten a lot of external recognition in the last couple of years on sustainability, and I think this last year we were number 3 in Barron's list of top 100 most sustainable companies. So glad to be here today, and thank you.
Thank you, Darlene. Zach Serber is the co-founder and the CSO of Zymogen. Welcome, Zach.
Thanks, John. Hello, everyone. In 2013, I, along with 2 others, founded Zymogen, a reasonably large synthetic biology company that has kept a pretty low profile to date. We built our business on the premise of using automation and data science to be able to optimize cellular phenotypes in profoundly faster, with less risk and less cost than ever before. And we built a business catering to others already engaged in biomanufacturing to improve their microbial catalysts. In 2019, the $1 billionth of goods sold using one of our microbes was transacted. So over $1 billion, well over $1 billion now, of products sold with our microbes. And now we are turning our attention very much to developing our own products. We partner with nature to create new products, new to the world, that solve unmet market needs. And we work in a number of sectors, and we're launching our first product in 2020, and it is in personal electronics. We are very much focused on using the exquisite chemistries available to living systems to create new monomers and polymers with distinctive properties that serve to outcompete traditional petroleum-sourced sources of those not equivalent chemicals, different chemicals, but similar substance categories. And today I'm hoping to talk about how Zymogen has seen both the role of automation and the role of data science in bringing about a change in the bioeconomy by reducing the effort required and the risk associated with bringing a new product to market. Back when I got started in this field in 2007 and was at Amyris, there was more or less a one company, one product paradigm. It took teams of hundreds to bring a product to market. These days, at companies like Zymogen, that same team can pursue a portfolio of products in parallel, scores of product, and that's all made possible because of the great advances that have happened in the last 10 years around automation and data science.
Excellent, thank you, Zach. And Jay Kiesling is the CEO of JBay and professor here at Cal. Jay.
Hi, John, thanks for the introduction. And I'm also a senior faculty scientist at Lawrence Berkeley National Lab, just up the hill.
How could I forget?
Yeah, how could you forget?
all of the many titles.
In fact, this morning, I apologize for not being here. I was teaching across the street, teaching chemical process control. My laboratory for the last 28 years here at Berkeley has worked on engineering microbes to produce chemicals. Some of these are pharmaceuticals, like an anti-malarial drug called artemisinin, to fuel molecules, all the way to some other kinds of molecules, like cannabinoids. And along the way, we've launched several companies from the research in my lab, and many of my students have gone out and launched their own companies. About a dozen companies so far have come out of the science from my laboratory. So I'd like to think that I'm also helping create or nurture innovators that then go out and start their own companies here in the Bay Area.
Absolutely. Wonderful. And I don't think the industry would exist without you, Jay. So thank you. I was in Berlin just after Christmas visiting Bayer, and in particular the part of Bayer called Leaps by Bayer. And who here has heard of Leaps by Bayer before? So Leaps by Bayer is the venture arm of Bayer, and it's called Leaps because it's like one giant leap, but they have 10 giant leaps of things that they want to disrupt over the next few years with their investments. And they're things that range from protein for food production to cancer immunotherapy. There's a long list of 9 applications. And when you get to the 10th, it says something along the lines of apply digital technologies for truly transformational business. And I found it really interesting because all 9 are applications, and then this 10th one is a very broad, we need to get more digital in our business. And I thought it was interesting that it was kind of snuck in there at number 10. Zach, I think Zymogen has been living and breathing this since the beginning of the company. You're only 7 years old, as you said. Darlene, for a company like Agilent, clearly you've spun out of digital, being HP. But can you talk a little bit about the impact of digital technologies on Agilent? Or is there a need for Agilent to transform?
Everybody has to transform. And digital is hugely important in Agilent. And of course, the HP roots are 20 years ago now. But it's really about being able to achieve the business results and contribution that we want to be able to do. And so we think, in a company as large as Agilent, we think about digital along kind of 3 different vectors. So there's the customer experience piece, there's the internal operations, and then there's products and solutions. And probably not surprisingly, I think the most interesting is products and solutions, but certainly it's really important in all the different areas. And so it kind of touches the whole company. Products-wise, we think about the science and the economics, or kind of operational efficiency and productivity of the laboratory as kind of 2 parallel tracks that obviously intersect. And so one of the places in science where it is incredibly important is in pathology. And tissue, we analyze, probably 1/3 of the world's tissue biopsy fine needle aspirate samples in terms of cancer diagnostics. And all of those today are still evaluated, these tissue stains, by an eye and on a microscope. And the complexity of these samples is really getting beyond what the eye can determine. And so this whole area of digital pathology is so important, because with all that we're learning about tumor biology, there's many different cell types and a much more complex tumor microenvironment that is needed in order to really inform the whole molecular pathology side of things. And so with that, AI is really the only way to integrate all of that tumor biology knowledge into the next generation of diagnostics. So that's a really, really big area for us, and I think incredibly exciting in terms of how we're going to advance areas like immuno-oncology in the next few years with that.
Thank you, Darlene. Jay, if you look at your lab at J-Bay, if you could just describe it briefly in terms of how many graduate students, how many postdocs, how many researchers you have, and then if you could describe the transition that's gone on over the last 10 years. Many of us here have been in the lab pipetting. Is that still common? Do we see automation equipment? There are now benchtop liquid handlers that cost just a few $1,000. Does each one of your grad students have those instead? Can you help us on the journey of
Sure.
what the impact of these technologies has been on your lab?
Sure. Thanks, John. So the Joint Bioenergy Institute is funded by the U.S. Department of Energy, and our goal is to transform cellulosic biomass into biofuels and other products that might be made from petroleum, or even products that you could never make from petroleum. And we've been around for about a dozen years. But we're a basic science research institute, so we get technologies out to industry. We have about 200 people working in the J-Bay environment on the 4th floor of Emory Station East. About 130 of those are paid through the J-Bay grant, and we have a dozen partners, 6 from national labs, 6 universities. Let me give you some perspective. So 28 years ago when I started in this field, there was no World Wide Web. There were no sequenced organisms, practically. Maybe there was 1 or 2. Things have changed dramatically. If you can imagine doing metabolic engineering on an organism when you don't even have PCR to use, it was really the dark ages. And where we've come now is tremendous and amazing, right? Digital infects the entire institute in some ways, in the entire field, and has allowed us to do things that are really exciting and fun and open up new frontiers, like you heard about from Zach and Zymogen. We still have people using pipettes at J-Bay, and I anticipate that will happen and continue for some time, because there's always the one-off experiment that you might want to do, especially in academics. But automation is pervasive. It's throughout J-Bay and through all of the companies that have come out of J-Bay. And it just allows you to do so much more, so much faster, in such higher throughput. You can screen through variants of enzymes that would have taken years before. You can do in a week or 2. Organisms can be engineered, and we can screen through them very rapidly and hopefully hone in on one or a few that are going to be much better. The academic environment is interesting and unique from some of the corporate platforms, because we're in an academic environment. We're really about creating knowledge and teaching, and so our platforms are very broad. We have many organisms in J-Bay that we're working on engineering, from plants all the way to fungi to bacteria. And we have many different products that we're trying to make. You heard a talk, I think, today from John Mello and one from Eric Steen, talking about the work in those companies. And they are phenomenal, but they can get very far because they're working on a smaller number of platforms and a smaller number of chemicals, trying to get to very high levels of production. But that impacts how you do automation, because at J-Bay we have to have a very flexible automation platform that'll work for both plants as well as for fungi as well as for bacteria. And that's very different than if you're operating an automation platform that works on one or a couple of hosts.
Zach, I believe the Zymogen platform is pretty flexible. I think you've got a number of hosts that you also work with. Can you describe a little bit about what goes on, and maybe describe, if you could tell everybody what percentage of the company are computational people, what percentage are biological people, what percentage are automation people, how does that look, and maybe also then comment on the changes that you've seen from grad school to Amyris to Zymogen and what you're seeing in the future.
Sure, and let me start by talking about the composition of the company, because I think it might surprise some people. So we are a company a little over 800 now, and of roughly 600 technically oriented folks, maybe 180 are have a biology background.
Wow.
So it is a minority. Clearly, we depend upon biology as our core value proposition, but the ways in which we go about imbuing cells with the capacity to make a new product and then optimizing it may not very much resemble as it's done elsewhere.
So we're automating ourselves out of our jobs.
Well, it changes the scope of what you set out to accomplish. As I mentioned before, there used to be this one company, one product paradigm in the synthetic biology startup community, and that's given way to a more portfolio approach. And actually, to answer one of your earlier questions, we have worked in scores of different microbes, filamentous fungi, yeast, bacteria, largely driven by what our customers were already working in. So the bioeconomy, biomanufacturing as it exists today, much of it grew up around what a microbe natively produced, and then it was coaxed into making more until you got commercial scales. And so there's an exotic collection of microbes currently employed in large-scale fermentation around the world. And we've had to adapt our capabilities to be flexible to accommodate all comers. We didn't have the luxury of being able to say, oh, we only want to work in Cerevisiae, because that would have limited our addressable market very much. And so we've taken a very much all-comers attitude to it, and it's definitely affected how we think about modularizing and automating biology. For example, transformation, introducing foreign DNA into a cell, involves getting it across the membranes and the cell walls, if there are any, and integrated into the chromosome. That there are roughly half a dozen different bona fide techniques for doing that, and we have automated all of them, and we use design of experiment and automation when we're considering a new host to work with to optimize our protocols against that new host, and we'll try all 6. We won't take conventional wisdom for granted that the best method must be conjugation, for example. We will explore all of them, and then we will also use design of experiment to parameterize it using the robots to explore variable space efficiently to find the optimal protocol to get the highest efficiencies of transformations. And all this is turned into an SOP, a process that can be run by non-PhDs. So we have a large number of technicians at Zymogen who run the protocols, who go through a series of steps when it comes to, for example, onboarding a new host. The largest other group of technical experts outside of biologists are software programmers and data scientists. Our tech group is large, about the same size as our biology group, and the remainder is made up of automation experts. We have a healthy number of chemists now. We have a group of about 75 chemists, all working on different applications of biomolecules into different sectors of the economy. As to how things have changed, it's staggering to think. When I was in my final year of my postdoc in 2006, 2007, I ordered oligos to synthesize my own gene, doing the assembly PCRs myself. And that is just so far-fetched these days that anyone would have to ever go to that trouble. And even that was a pretty big step above and what had been the case where you had to go find the researcher somewhere else out in the world that happened to have the gene sequence that you wanted and ask them to send it to you so you could PCR it up. So we've come a very far away from that. I mean, right now, one of our assets is this extensively well-sequenced metagenomic library that we created from, well, we have several, but from different ecosystems. And the abundance of genetic diversity in there is a starting point for a lot of our innovation. We have a digital collection of genes that's about 5 times larger than that in the public databases, wow, which is our go-to source for enzymatic diversity. And rather than try, as we have in the past as a community, try to think one's way to the best starting point, let us employ the robotics and our capabilities to empirically evaluate which is the best starting point. Let's then feed those data back into a central database, which then can be mined for patterns so that when it comes next time to hunt for the right enzyme, we're using our prior experiences to down-select bioinformatically in the most sensible fashion.
And one of your early hires was CTO Aaron Kimball. Aaron was from Cloudera. So you hired from the tech industry into one of the early hires.
One of the experiences I think many people at mature companies have when they're trying to digitize their work is that a huge effort must be placed upon what I refer to as plumbing. That the data in mature companies that have been in the market for a long time, it exists, but it's in disparate locations. It's not organized in any central repository. It's very hard to unleash the potential of data science and the experts in machine learning and whatnot on the data sets because they're so disorganized. They're so, I've heard many cases where a company gets a little ahead of itself, will hire a team of data scientists and say, okay, now go to it. And they spend their first year frustrated because all they're doing is plumbing. They're making sure that the data are actually available to them to do the experiments, the machine learning experiments that they wish. We anticipated that, and Aaron Kimball came from a cloud-based scalable data infrastructure background. And his task was to make sure that we had a central repository for all kinds of data. And we did not yet, at that point, fully appreciate all the ways in which data was going to help us. So we took a very generous attitude. We wanted a flexible, scalable, expandable database that could take data of all sorts, whether it be a genome sequence or an image file or a design strategy. All of that could be compiled, and then later, and it took us a couple of years to acquire the data that was robust enough to start to unleash the machine learning, but it was that much easier because we had anticipated the data structure problem.
I remember 10 years ago when I was in graduate school, there were data biologists and there were wet lab biologists, and neither of the two shall meet. That would be pretty rare that they would meet. I'm excited to see what the new graduates are coming into the workplace with the skills that they have. Can you maybe comment on that? Are you seeing, presumably you're seeing these wonderful hybrids now emerge of people who have both of those?
Oh yes, and frankly, there are also certain schools that are better at this than others that make quantitative training, computer science and programming training integral to their bio-based programs. So I've laid out a staffing model which neatly puts people in buckets, but in fact it's more complicated than that, as you suggest. There are definitely hybrids, people who straddle, and in fact, for good company culture in a place where you are trying to bring together technical experts from so many different backgrounds, it's critical to seed that community with people who can translate between these different arenas productively.
Jay, Darlene, any comments on workforce development?
Oh, I was going to actually make a comment on automation.
Yeah, sure.
Which is, automation is huge in all of our manufacturing as well. And one of the more exciting examples is we just built a GMP facility for large-scale manufacturing of RNA, or oligonucleotide in general, but RNA in particular therapeutics. And it's amazing how much automation and intelligence has been incorporated every place it can be in this, when you start from scratch. It's really exciting to be able to do that. But beyond large-scale manufacturing, the kinds of things we're talking about here with Zymogen, and is that one other aspect of automation, in addition to being able to have the efficiency and the throughput, as Jay and Zach were talking about, is even on a single lab bench, adding automation can address improving reproducibility and accuracy whenever there's any meaningful level of sample handling and sample prep that's going on in that workflow. And I was really amazed when we first put like the AsciiMap Bravo in front of the mass spec and saw, literally 2- and 3-fold improvements in that reproducibility, even with skilled users, but there's still differences between users and so forth. And so small scale, there's a lot of value in manufacturing. You obviously just have to invest what makes sense for the cause. But like as an example now, we've just introduced front-end automation for NGS workflows in terms of library prep and target enrichment with, again, full automation. And I think we're going to see similar types of enablings of, let's just say when you're doing hard experiments, the more you can reduce the sources of error, the better.
So I'm going to be opening it up to audience Q&A in a couple of minutes, so have your questions ready. I'm curious, which parts of the engineering biology workflow do you all think is going to be the last to be automated, or the last to be having AI and machine learning applied to them? Where do you think the last holdouts for the human brain having a productive function in the lab are going to be? Jay, you're smiling.
Well, I was going to say that, but I might offend Darlene here, that one of our pressure points is analytics. Mm-hmm. And no offense to Agilent, they make awesome equipment. So do a lot of other manufacturers. But I think we're going to get to a point, or we're finding, where we can engineer cells now faster than what a lot of our analytical equipment can keep up with. And it depends on what your measurement is, but I think there's lots of potential in the analytic workflow space for high throughputness, and clearly, learning from our designs and getting that incorporated into the next designs, learning from the failures, the successes, and getting that in. I think there's still a huge space there for innovation.
So Jay, I agree with you entirely. So relax.
I'm glad I didn't offend you.
But no, I mean, you're absolutely right. And, you know, we're all part of this cycle. And I think that design and build have really been enabled in a big way through automation. And when you think about, you know, the test part, I think, you know, the throughput is there. It's really about getting the meaning out of the data, being able to bring all that phenotypic information together and then integrate it back into design. And that is, I think, by far the ripest opportunity for AI in terms of both addressing that data, but also being able to, in real time, incorporate the decision making into design.
Yeah.
So that's the bottleneck that's ripe for, I think, taking a big step forward.
My answer is a little different. I think Zymogen, we're getting pretty close to a closed loop system. Wow. Where the designs are algorithmic, the data are collected. There's human interfaces. It's not a zero-touch environment, but the data are collected automatically, fed back into improving algorithms that do subsequent designs. We spend a lot of time thinking about how to hone those algorithms, how to hone the manner in which the designs are generated, and we do a lot of A/B testing and the equivalent to compare different algorithmic types. The place where I think we have a healthy amount of human involvement and will for some time to come is to figure out how to plug this increasingly autonomous engine into the economy. Where are the best and biggest opportunities for biomanufacturing, for the bioeconomy to participate, often in places where we may not have yet a track record? I think there's a lot of, especially when this becomes more routine and the barrier to entry is lower, the cost and the time and the risk is low, it encourages us collectively to be expansive in our thinking about the ways in which the bioeconomy can impact the real economy and the rest of the real economy in ways that it hasn't before. And that requires a lot of pavement pounding and interfacing with the existing players in the market, coming to understand. Some of the unmet market needs are so long held and profound that people even fail to recognize them as unmet needs any longer. They've had to exist or coexist with them for so long, and trying to suss those out is still a very human endeavor.
So good sales and BD people then is one of the last last things that will be automated.
Yeah.
Any additional comments?
I think there's still a lot of opportunity also in scale up. We can get things to work really well, maybe at the bench scale, and going to scale up, I think there's a lot of work that can still be done, a lot of innovation that can be done in getting things from bench scale to full scale.
So can you elaborate a little bit more on that, Jay? What maybe you could talk about the work that the ADPDU has done, and that was set up specifically to try to get through this valley of death around scale up. What are still some of the big bottlenecks, or if we could have another 10 ADPDUs around the country, what would you have them working on?
So for those of you who don't know about the ADPDU, it's the Advanced Biofuels and Bioproducts Demonstration Unit. It's part of Lawrence Berkeley National Laboratory, funded by the Department of Energy. And it's this idea that you need a very flexible facility that any number of small companies can come into. And with their process, they can scale up those processes to larger and larger scale, something that maybe for a very early stage company, they need the data on getting to, say, a 300-liter fermenter. And I think that facility is quite unique in being very flexible, being able to work with many different pre-treatment processes as well as many different fermentation processes. And in fact, the EBRC just had a meeting in the Houston airport on Friday where we were talking about kind of the lack of scale-up facilities and manufacturing facilities that can be leased by companies here in the U.S. and how difficult it is to get access to those. You know, there are a number of almost case studies of companies where they've had challenges, where they've engineered an organism, gotten it to work really well, and then they go to the full-scale production facility, and something happens along the way, and it doesn't scale up. And so I think there's still a lot of science that can be done in this area. and automation that can be done to maybe smooth out those transitions going from small scale to large scale.
I want to throw it out to questions, so please put your hand up. Great, we have a lot of questions, so we can have a mic over here, and if there's another one there. Oh, we have cool QB questions. Okay.
Ready, John?
Matt, over to you.
Thanks, John. Jay, this question's for you, really, and I've been thinking about this question for quite a while. So we're kind of at this second wave of companies getting to scale now, and in some ways that makes you kind of grandpa.
Matt,
Thank you very much.
founder of Bay Bio, if you could just introduce yourself.
Thanks, John. So I'm Matt Gardner, and actually worked with Jay very directly for a few years, so that's why I felt comfortable calling him Grandpappy.
Yeah, thanks.
So Jay, what would you say has changed about making it easier for entrepreneurs now as opposed to 10, 12 years ago?
Well, first of all, I'm in Berkeley, in the Bay Area, and so what I'm about to say may not apply to other places in the country, but what I've noticed is there are so many more resources for young any entrepreneur, but in particular undergraduates and graduates and postdocs here at Berkeley, and I know the same thing is true at Stanford. So many resources, classes to teach them, so many more possibilities of getting funding in interesting ways. And they're getting exposed now in these environments. I know at JBay, where we kind of sit in this cradle of synthetic biology in Emeryville, that they get exposed to this community every day, and it's really fantastic. And the same is true here on the UC Berkeley campus. And so I think it's just the access to the education, to the resources, monetary and non-monetary, the incubators, all of those things together has, I think, made a much richer and interesting environment. And if you track SynBioBeta and all the work they've done to track this industry, you see how it's grown tremendously over the last decade. It's really amazing.
Yes.
Yes, my name's Paul Bryan. I'm a senior scientist at Sandia at the Livermore site just down the road. And my first time talking to a foam cube. But this is not going to come as a surprise to Jay because he's heard it from me many, many times before. And he had a perfect pitch to hit with scale up, but sorry, Jay, you swung and missed. To biologists, far too often, success with a product means they have a molecule at a modest titer in an ocean of water. Possibly it's a molecule that likes to ionize, and there may be other molecules in that same solution, and that's not what you want for a product. So you have to do separation, purification, recovery, possibly formulation into a particular crystal or something for pharmacological type materials. And so I think the path from basic science to a real commercial product has to take into account that recovery and purification, and also in some cases further conversion of that molecule by more conventional synthetic organic chemistry.
Did you want a response, or was that just a statement?
I'd like a response. How do we move that forward along with the biological side?
Yeah, so yes, you're absolutely right that downstream processing is critical. And in some time in the future, I think we're going to be designing organisms de novo, where we know the substrate that we have available, we know what product we have available, we know all the downstream processing conditions, and we'll just build the microbe suited so that we have it in an ideal situation for downstream processing, so we can include that all together. And Zach's probably going to tell you that they have that already. They probably also have the full suite of all the known synthetic organic chemistry to decorate whatever molecule comes out of the organism. But those are the kinds of things that are coming online now, and I think that we'll see more and more of in the future.
And to build on that, I think I completely agree with the sentiment. I mean, downstream purification is essential and often overlooked until too late. I also think there's a lot of room for better partnership between the eventual scale-up process, both in fermentation and DSP, and the choices one makes on the biological side. So one of the advantages we've got by having worked in so many hosts is we can choose a host best suited to the product class we're producing and the eventual process we're going to be using for purification. And the choices don't end there. So if you have a co-product which is making DSP purification more tricky, adding an expensive step, what can you do on a genetic level to remove the presence of that co-product to simplify and cheapen, make less expensive the downstream purification? Having a team that's accustomed to working through these problems together and to start with the end in mind is critical to both going quickly and to ending up with a viable low-cost product.
Now, a big part of this scale-up problem and the downstream processing problem is that a lot of this is competitive amongst various industry members. Darlene, can you think of other industries or other areas where there's been pre-competitive collaboration amongst industries to try to solve some of these big, gnarly problems?
Well, I mean, I think that obviously, from the pre-competitive, the semiconductors, of course, been a great example from many years ago. But I'm seeing more and more within synthetic biology in terms of collaboration and a lot of the biofoundry work that's being done and led out of the not-for-profit space. So I think there is that today, and the scale of what needs to happen is so large that there's plenty of room to identify some fundamental areas where people are collaborating, industry, academic, and government, with that.
Great.
Yeah.
If you have another question, please put your hand up high, and we'll get a cube to you. Yes.
Hi. I'm Dave Humbert, and I went to Cal along with Jay. And just thinking of when I was at Cal, I did a simulation project. So I found myself learning C++ in addition to chemical engineering and graduate thermo, et cetera. So I guess thinking about how younger people are coming up today, for an adjuvant for a Zymogen, you could presumably hire data scientists, but I get you do. But are there pain points, things you're having to re-educate your new employees on, and then what could chemical engineering at Cal, for example, pre-educate them on for data science techniques, et cetera?
So I would just offer that in terms of the education side in chemical engineering, the more folks can feel comfortable in a multidisciplinary environment and be able to communicate across science and engineering, that's incredibly beneficial to where so much of the forefront research is going and where the new opportunities are at the interfaces across disciplines. So that would be the place. And from what I know about engineering and chemistry at UC, I think that it's already happening. The other piece that we always end up teaching, is interesting to reflect back on Zach's comments, but it's the business side and realizing that it's not just about great technology, but that you really want to solve a customer's problem and make a difference that's going to matter. And having a better understanding of business models and how to create value and differentiation, as part of it. But people learn that along the way, but that comes back to the judgment and the parts that are not part of the, design, test, and learn.
Mark.
Hi, I'm Mark Salet from SLAC and Stanford. If we're talking about digitizing biology, at what point does the digital information really become the raw material, and we're talking about an extractive industry, albeit a pretty innocuous extraction? Who owns the data? Zach, you talked about proprietary data from some interesting metagenomes. There's a genome project for the UK that's been funded by the Wellcome Trust. They're trying to sequence all the organisms in the United Kingdom. There's been talk of an American genome project, sequencing everything in the continental United States. Who knows about Hawaii and Alaska? They must be thinking about that. what's the role for public data and the availability of that to feed our biomanufacturing in the future? And can you contrast that with private data?
I guess that's for me.
I think it is.
Yeah. Sure. Yeah, that's it. So we are agnostic as to our sourcing of data, and we rely on public data as well. And there are certainly times when we will find a sequence of interest in the public domain that we don't have in the freezer, but we will go out and synthesize. When we have it in our own proprietary database, we actually have frozen stocks of it as well, and we can PCR it out. It's a little faster and cheaper. That's not the root of your question. We also heavily rely on the public community to give us a framework for taking the data science-driven approach. Design space in this field is immense. Everything that's alive today and everything that has ever been alive was the way it was because of its sequence of DNA, obviously, and many gazillions of things that haven't ever existed could also be encoded by DNA. The choice, the down selection, if you will, from all of that design space into something that you can both handle both computationally and then empirically with liquid handling robots requires some framework for down selection. And were we to be where we are today with the computational power and the robotics power, but without the benefit of the framework of biological understanding provided by over a century of fundamental research, we'd be nowhere. There'd be no way to go efficiently from infinite design space to something that's actually liable to matter. I mean, for example, Zymogen, when we contemplate the genome and we consider it a global optimization problem, we anchor our algorithms according to ORFs, to open reading frames. We don't just treat it, we don't go down to the ATCG level and consider all those possibilities because that level of detail betrays our ability to manipulate it at any kind of reasonable scale. So we have to lean on gene annotation and things like that, where we're pulling resources and know-how from the public domain all the time. So there's a very fruitful intrinsic reliance of companies like Zymogen on the public domain for what we do.
And do you contribute back to the public domain?
Depends on how you define public domain.
Okay, that's all right.
Yeah, so we have been talking internally, but we haven't done anything yet about our proprietary metagenomic sequences. And there are things in there that are of potential value and interest to people outside of our business model. And yeah, perhaps we should.
Yeah. Sorry to interject that.
Any additional questions? Yeah, we have time for a couple more.
Seth Snyder, Idaho National Lab. We operate the National User Facility, the front end for much of the work here of the feedstock user facility. My question actually is tying some of the stuff that Darlene and Zach said. You know, you talked about the digital side of it and how instead of going from one company, one product, sink or swim, to this opens up infinite possibilities, and you were just leading to that and how you make decisions on offs versus going down to individual genes. And Darlene, you pointed out business models. You put a room full of scientists and say we have an infinite number of possibilities. You have a lot of risk that you will spend an infinite amount of time because there's an infinite number of rat holes. How do you take scientists who have a large ego and drive them and say, this is what we want to get done, how do we, and make decisions to end projects or to focus on projects? It's a big challenge to be really successful.
It's a really great question. And you know, one of the things I've had the privilege of doing for a large part of my career has been leading the research labs. And it's an amazingly talented group. And all ideas, many of the ideas are good, not all. But to actually decide where do you put the investment. And I think that the more that one can explain where there's sort of two sides to it. One is being really clear on what you are trying to do, and so that and being very transparent so that people understand what the criteria are as compared to what's just the best technical solution. So I think that's one side of it. And the more you can explain and acknowledge that you've thought about what they think you should be thinking about, the better, as compared to thinking that you're ignoring something that's really important. That's something that I think all of us, many of us in the room as technical folks, really care about. But I also feel really strongly that sometimes you have to just get your hands wet and do some things. And that you don't want to dismiss something that maybe isn't as quite like what you think it should be, but you have to encourage that curiosity. And being able to start a new area and start a kind of a pre-investigation, if you want to call it, maybe spend 6 or 9 months with 1 or 2 people exploring an area, and being, not necessarily trying to answer the easy questions, but to really hone in on what is the important question that we'd want to understand to figure out whether there's really a there. And so I think they're having a say yes versus say no attitude to some of these things is also really important. And a lot of that is based on, I think it's based on gut reaction. And I will say in a very respectful way, listening to some of the folks that have really been successful in the past at those types of judgments, and really valuing that, even if you're not sure. So it's kind of like it's a bimodal answer, but maybe hope that helps.
Thank you very much. So we are at time. I will finish the last part of my story. If you are familiar with the California wildfires in Paradise last year, just about a couple of hours north of here, there's a big problem in a lot of the burnt trees that are left behind from these wildfires, and they're not stable, they're not safe to keep them around, they have to be pulled up. So the company that has the contract to pull these trees up is called Recology. If you're in the Bay Area, maybe Recology is your recycling and trash provider. So next week, I was telling Mary, I'm going on a landfill tour with the COO of Recology to look at some of these landfill sites, which are filling up or piling up with these trees. The current plan is to chip them, truck them to the coast, put them on a ship them to Korea and Japan, and burn them for energy. Yes, that's what I said. So I've been floating 2 ideas. One is a wonderful program that Blake Simmons has just got funded, $6.5 million for gasification and mobile biorefineries. Very, very exciting program that Blake has just got funded. And that, of course, could feed in as a syngas into any one of these manufacturing fermentation companies like Zymogen, or if Zymogen win the fermentation business, or LanzaTech, I was speaking earlier with Sean. But the second opportunity, who here is familiar with Ecovative in upstate New York? So Ecovative is a mycelium-based materials company. So they're making packaging material, they're making leather. They actually already have a partnership with a company called Paradise Packing Company up in Paradise. But I've long had this vision that we can create giant monuments. So I was on the phone with the CEO, Evan Bayer, last night, who's going to introduce me to the packaging company, and we're going to take Recology there. But Evan said, I've never made a dome. I just got back from India last week and saw the Taj Mahal. He said, I've never made a dome. So I said, well, let's try to get Recology together with Ecovative, and you will have the Taj Mahal built from mushrooms in paradise. And with that, I will end and thank all of our wonderful panelists. Thank you.
Wow, that was exciting. The bioeconomy is not part of the real economy. Zach, come on. Earlier today, in his opening remarks, Director Witherell made the point that the National Academies just came out with a new report, Safeguarding the Bioeconomy. And for the first time ever, the US bioeconomy has been measured. $959 billion, almost $1 trillion. It is a very big part of our real economy. All right. I talked a little earlier about the innovation that this event, the biomanufacturing event, brought to the InnovationXLab series as IP portfolios. We have another one, and that is that we are the first XLab to bring you a focus on ethics. Now that probably shouldn't surprise you when you work with living organisms, self-replicating things that are alive, it's important to think about what we're doing and how we're doing it. So our next speaker is well established in this field. Alta Sharo is the Warren P. Knowles Professor of Law and Bioethics at the University of Wisconsin at Madison. Professor Sharo is well known for her national service, as she is for her scholarship and teaching on law and policy related to such issues as environmental protection, medical genetics, stem cell research, and research ethics. She served on such boards as the former American Association of Bioethics, as well as the Ethics Advisory Board of the Howard Hughes Medical Institute. She's also served as a consultant to the National Academy of Sciences Institute of Medicine. In 2005, which is about the time I think I met her, she helped draft the National Academy's guidelines for embryonic stem cell research, and a year later was appointed to co-chair the National Academy's Human Embryonic Stem Cell Research Advisory Committee. She was a member of the National Academy's Human Gene Editing Initiative and co-chaired its committee charged with making recommendations on the use of gene editing for both somatic and germline changes in humans. She's currently a member of the World Health Organization's Expert Advisory Committee on Global Governance of Genome Editing. Please join me in welcoming Professor Sharo to the XLab stage.
First, thank you very much for that rather over-the-top introduction.
Yeah, you can pay me later.
Excellent. Does somebody have the slide clicker? Got it. Thanks. So there are an enormous number of, let's see, which goes forward? This one? Green. Green? Great. Ah, good. Okay. Oh, I won't even be able to read my own slides. So there's an enormous number of applications, particularly in genome editing, ranging from crops and animals to basic science, discovery science, stem cells, and of course, what you've all heard about, most probably in the newspapers at least, which is germline editing for designer children. I was asked to talk a little bit about kind of comparative thinking between the EU and the United States. So I'm going to talk about a very limited range of things because there just isn't time for all of them. So the first one I wanted to mention has to do with biobanking and the use of stored tissues and the data from the stored tissues, which seems quite on point considering what we just talked about in the previous panel. Most of you may know, but for those of you that don't, the Europeans have a rather scattered system for regulating this, a lot of variation among countries, but there are a few new things on the horizon, including the data protection law, that are now beginning to cause the countries to begin to converge on the basic management of the biobanks. And in many ways, they're very similar to the US in the idea of voluntary donation, the distinction between anonymous versus identifiable tissue, the notion that people have some degree of control over the use of their tissues. These things are very common between the US and the EU. But the other thing that's very common are the areas that are still very much in flux. For example, there is really a very, very poor literature thinking through situations in which tissues are anonymized, the individuals cannot really be harmed themselves by any kind of result from it, and yet might have objected to the underlying research that's being used for. Could be research on eugenics, for example. And we in the United States and in many places in Europe treat anonymized tissue as if people have no control over its use. And this has still not been very well thought out because, at least in the US context, we don't think of anonymized tissue as representing a human subject at all, so it evades all of our rules. The use of consented tissue, but for unanticipated research. Increasingly, people are using broad consents, asking people to say, yes, you may use my tissue for anything in the future. But the Europeans are actually beginning to think a little bit more, in a little more nuanced way about this. And even in the US, there's some concern that this is basically a consent to not be asked to give consent. So as long as we insist on this notion that the tissues are really something that individuals should control as opposed to being a public resource, which is going to raise questions, as you just did, about public duties, we're going to run into this question about whether people, in fact, can waive the right that the law has given them to control the use of their tissue. The use of consented tissue by for-profit entities. A lot of people donate tissue thinking it's all going to go to science, and science is all white hat, and it's all people scurrying around in laboratories being paid very poorly. I mean, for those of you that remember Sinclair Lewis, it's the Martin Arrowsmith kind of image of the scientist. When in fact we know that often, in this room particularly, there's a for-profit entity that's going to take advantage of this, whether it was put into a private database through a pharmaceutical company or it was put into a public database. We have experienced at least one time a really significant public backlash when people who donate something thinking it's altruistic discover somebody else is making money. And I'm thinking about the incidents in Southern California when families that had donated tissues like bone and skin from their deceased relatives, thinking it would be used immediately for some kind of treatment, like a skin graft, discovered that in fact some of the bone was being machined by intermediaries into medical devices that could then be sold for a profit. This is not something I think is unethical, but it is something where I think we haven't yet thought through how we want to manage it and how we want to talk to the public about it. And then finally, this is now a major concern in both the US and the EU, and that is what to do when you discover something that you didn't expect to discover. You know, do you go back to the individual? Do you go back to some intermediary like a physician? Do you even have the capacity to go back? More problematically, do you have enough information to make it make sense to go back? Do you give people information about things where we don't understand the significance of a variant that we found, where we suspect that it may be associated with something, but we don't know if there's a causal relationship? Do you wait until there's some kind of proven relationship? And having solved all of that, what happens when you've now done that, and it was 1990, but now it's 2020, you still have those samples? Should you be re-examining old samples looking for the same things that we're discovering now that we wouldn't have thought to look for in the past? So for all of you that are working with biobanks or working with data from biobanks, it's worth at least keeping these things in mind because they are going to be solved by politicians and regulators. So it's important to understand what the dynamics are in terms of your use, what you need, what you don't need, and what kind of protections you might want to advocate for when you're working out what the new rules are going to be. They are going to be coming down the pike. Now, another area of EU and US comparison that we frequently see has to do with the management of genetically engineered animals and plants. And as was mentioned before the lunch during the panel that Larissa Rudenko was moderating on foods, there were a number of people who highlighted that the EU has a much less generous attitude toward engineered foods. Well, you know, this is something that is an old kind of phenomenon in Europe because, as you know, they regulate by the process and not by the end product, which is the US approach to these things. Now, one of the more problematic things that has happened is that the European Court has determined that gene-edited plants and animals are going to be treated the same way as the old transgenics were. Now, there's two things that were interesting about that particular opinion, aside from how frustrating it may be for some people to see that edited things are going to become subject to the same kind of restrictive rules. The first, I'm going to go into it next, has to do with the way in which they're interpreting the so-called precautionary principle. But the second that's rather interesting is where they talk about how gene editing or genome editing actually allows you to produce modified varieties at a rate out of all proportion to those resulting from the application of conventional methods. This is a different way of expressing the concern. I like to talk about something called the ethics of efficiency, that things that are hard to do are in a sense self-limiting. When sex might always result in a pregnancy, then it is self-limiting in that people who fear an unwanted pregnancy have a reason to not engage in sex. And yet, when the birth control pill comes around and you now have an efficient way of preventing pregnancy, we suddenly see an explosion in sexual activity outside the confines of marriage, the 1960s, for those that remember it, or those who have parents who remember it. It changes the nature of human relations by virtue of the fact that something has now become efficient enough that the other self-limiting factors are no longer present. And I think that's really part of what's driving the court here, is really a concern more about efficiency and scale than the actual question biologically of what is or is not the essential nature of this organism that they're discussing. And since a lot of the discussion today has been about scaling up, it is also something to just keep in mind, think through for yourself, when you scale up to this level, what other things are now going to change? And are those changes all good? And if not, what might be done in order to take advantage of the scaled up capacity, but at the same time, not get an unintended consequence, or at least anticipate it so you know how to react to it? The second part of that opinion is the one that references the precautionary principle. And there are different versions of this. I know that for many of us, particularly in the US, we think the precautionary principle basically means you can never do anything. It's not exactly that. It has different flavors. The first two flavors are, given that there's uncertainty, in one case, uncertainty means that you don't have to forego all regulation, and the second one is that uncertainty doesn't mean that you're not allowed to regulate. But the third one is the one that's actually the most problematic. And it's the version that says, if you have uncertainty, what you can do is shift the burden of proof and change the standard of proof. Now, the effect of this is profound because, as you know, you can never prove something has no risk at all. There is nothing that has no risk at all. And so shifting the burden might have the advantage of perhaps forcing the proponent of a technology who has the best access to information, including proprietary information, to do the hard work of digging up the risks and benefits. The manipulation of the standard of proof can render that effectively what people here tend to think about, which is precautionary principle means zero action. It is something that the EU has been talking about for a long time. In 2000, they enshrined it in language. And the language is kind of weird because it starts out talking about things that sound very reasonable, potential costs and benefits of action or non-action, for example, non-discrimination. But you'll notice in the red toward the bottom that they specifically talk about why it's okay to change the assignment of responsibility on the burden of proof. And that has probably been the point at which the precautionary principle has caused the most difficulty. Whereas in the US, we tend to actually have a fairly predictable notion of where that burden lies. If it's a drug, then the burden is going to lie on the pharmaceutical company that has to prove to the FDA that it's safe enough to be marketed. If it's a food that's already been generally recognized as safe, the burden is going to be on the government to prove that the food, in fact, is not safe before they can pull it from the market. So on a statutory basis, we've kind of clarified who has the burden and what order of events must take place. In Europe, it can be a little bit less predictable under some conditions. The other thing about the US that's different is that where genetically engineered plants and animals have been controversial in Europe, in part because of the question of whether they should be allowed at all, in the US, the shift has been more toward the question of consumer entitlement. The right to know, so to speak, which makes sense because in the United States, we tend to think in terms of rights and individualism and personal autonomy. And in Europe, the kind of terms of discussion are a little bit different. They're more public health oriented, more collective interest oriented. So you'll see the continued debate there being focused presumably on the health and safety with the underlying unspoken issue of the economic ramifications for their farm communities. Here, where again, it's really about capitalism and about agriculture, you'll see this kind of emphasis on labeling. How do we label? What do we define that we're going to label? So the arguments play out a little bit differently between the two jurisdictions. Now, the EU, of course, has a lot of people who've been very unhappy with its approach to genetically engineered organisms. And so you have articles like this appearing. There's so many of them, I can only clip one or two. But you hear about the new kinds of precautionary measures that must be taken in a field that are expensive. And then you have a Belgian startup that was going to be working with Brazil, but Brazil now is in a funny position if it wants to deal with the discrepancies in the trade rules and in the local domestic laws. So there's been a lot of pushback in Europe about this. And this is just from a few months ago, in which you now have coalitions forming. Here it was 117 research facilities that are calling for some kind of revision, particularly in light of the decision to now bring genetically edited organisms under the same rubric. And you know, I was struck this morning on the food panel at the way in which companies that have now been able to say our food is not made with genetically modified organisms represent in some way an emphasis on scientific research that has been driven by policy, which I think all of us would say is policy that is not actually grounded in good science. And so it's an interesting example of how bad policy can actually shift the emphasis of scientific research as well as product development. I'm not saying these companies are wrong to do it. There's a market question there. But it may not be the best way to set your scientific priorities by first having a law that makes no sense and then having people figure out how to evade the law, right? Not ideal. I want to talk next, because it's a short, they don't have a clock on me. Well, I don't have a clock I can see. I want to talk very briefly about the human applications of genome editing, particularly the heritable ones. Here we do really see some interesting differences, although the outcomes turn out to be the same. In much of Europe, there has been an agreement to sign on to something called the Oviedo Convention. It was something that was written by the Council of Europe, which is not exactly coincident with the European Union membership, but it encompasses a lot of the same countries. It basically says that you are not allowed to intentionally make heritable changes in the human genome. Oh, thank you. Oh, I got a manual, not even digital, manual. Very, very old-fashioned. And so for countries that have signed on to this, and a lot of the big countries have, the UK being an interesting exception, it is simply a criminal act to try to do what He Jiankui did in China. Now, I'm not saying he should have done that. In fact, that was an outrageous kind of example of premature use of science for no particularly good reason, without adequate controls or peer review. I also don't want to comment on whether a jail sentence is appropriate, but certainly the experiment should not have been done. But in these countries, it would already be criminal, whereas in China, it was a little unclear what the law said. I find it fascinating that even on this topic, which for many years, especially in Europe, was just a kind of a no-brainer. Of course, it should be a criminal act. It violates human dignity. It violates human rights. And in Germany, particularly because of its sensitivity to the history of eugenics under the Nazi era, a very, very tight and cramped attitude about anything having to do with genetics or embryos. We find the Max Planck Society publishing a statement that says, yeah, this is not ready, but you'll notice if you were to read through the very small print, they don't say it should never, ever be done. They simply say it's not ready now. And so they're cracking open the door the same way our National Academies Committee did in 2017. And it may be that it'll never be ready, because you may never be able to be confident enough about safety for something that has the potential to reverberate down generations. So it's hard to imagine how you would construct an oversight system that would be able to monitor for problems across generations like that with species that live as long as we do. But at least at a theoretical level, we're seeing now this kind of crack in the consensus in Europe about the kind of ethical defensibility of germline editing, at least at a theoretical level, question being, could you ever actually regulate it sufficiently to make it safe and effective enough to do it? The United States doesn't have total prohibitions the way the Europeans do. What we have instead are tricks by Congress. Some of you may remember that in the early 1980s, it was impossible to get federal funding to do work on human embryos, not because it was prohibited, but because there was a measure that said, if you want to do this work, you need to get an extra review by an ethics advisory board. And Presidents Reagan and then H.W. Bush simply chose not to appoint anybody to the board while leaving the requirement for its approval in the law. So effectively, you had a scientific protocol that would be slipped under a door into an empty room and simply wait, which it did for 12 years until a change of administration in '92. Well, a similar trick is being played right now. Germline editing would be treated as a biological drug because of the construct that's being used. And so what would happen is that it would have to go to the FDA. And we now have a provision that says the FDA is not allowed to spend any of its money, which means any of its staff time, on reviewing such an application. It would be a criminal act to do that experiment in the United States without getting prior approval under an IND for that from the FDA. And so effectively, it has made it criminal to do this in the United States, not by saying it's criminal, but by saying, oh, our agency can't review it to give you permission to do it. Right? This is actually not part of a permanent budget, but it's probably going to get rolled in and then get reenacted, because I can't imagine how we're going to get a majority of the people in Congress to stand up and say, yes, we should be funding germline editing. So I suspect that we're not going to see that kind of research here. at the clinical level. We'll see it coming out in the UK, Singapore, China, and other countries. I'm going to conclude simply by making a couple of very brief comments about where things are going. The Europeans have something called Horizon 2020, which has identified biotech as one of its areas of emphasis. It has noted that the precautionary principle has been controversial and has identified a counter principle called the innovation principle, where benefits are favored when you weigh them against risks. They have not concluded that they definitely want to go there, but they've started kind of a continent-wide debate on this. And it is part, I think, of a larger kind of effort. So I think what you can say in the end is that neither Europe nor the US has a completely consistent approach, nor can you say that on everything the Europeans are more cautious than us. They tend to vary, but certainly on some of the things that matter to you most in the area of agriculture, they are more cautious. In the area of biobanks, though, you may find that there's a lot of similarity and you won't run into quite the same problems. I'm going to skip over these two things, and whoops, sorry, how do I go back? Go back? Last comment, and I'll just leave this up here so you can read it. Some of these are the kinds of things that are going to be a matter for discussion for a while to come, and some of your companies may bump into it. Animal welfare every time you're editing an animal. The biodiversity question because of concerns about monoculture, gene drive and the environmental effects, and cross-border effects. We were talking about that over one of the breaks. The DARPA possibilities for the so-called super soldiers and other kinds of things that are part of the science fiction literature and now are becoming part of our real debates. But more than anything else, do we have a regulatory capacity? And do we have some venue by which the public can participate in these discussions? Last thought, and it goes directly to the question I think was thrown at Zymogen, which is, to the extent that the public is contributing through its tissues, through its medical records, through the data from the tissues, from its participation in clinical trials, and to the extent that we now have academic biology converging very, very strongly with business biology ever since the Bayh-Dole Act was enacted, how should we be thinking about the collective duty of the populace to contribute to the scientific effort, and what kind of reciprocal duties ought that create on the part of industry and academia to make sure that the results of their work are accessible in a fair way to all? Thanks very much.
Okay, so fascinating, provocative talk. Next time I'll have you come and do state versus federal rights, and we'll go into that. Really thought-provoking topic, and unfortunately we don't have time for Q&A, but she'll be around in the break session, which we're going to go to next. I want to thank Conagen for giving us the sponsorship activities and being such a supporter of the XLab. I also want to remind folks that we're still looking for a final few registrants for tours tomorrow. So if you want to take the opportunity to go to the front desk and register there for them, that would be greatly appreciated. Beat the rush, get your seat now. With that, we'll adjourn for the break. There are 2 feeding stations and drinking stations again, and we'll come back at, 3:30? Sure. Yes.
Why do I say that? It's because a speaker earlier today talked about the continuum versus a step change. Algae is really a step change from traditional terrestrial agriculture. And so this chart on the left gives you a plot of protein production per acre and oil production per acre, and I tried to capture as many of the advanced feedstocks as I could. If yours isn't on there, I apologize, but covering a lot of what today covers kind of through cellulosic ethanol or products from those kind of sugars all the way down to sugarcane ethanol. Those are big improvements over just some of the standard crops. And obviously you're making great improvements in terms of quality of products. Algae is really a different value proposition. We're talking about producing 30 to 40 times more oil and protein per acre than you are with traditional crops. And so it's really addressing the future much more than what can we do with what we've done so far. And that has tremendous impacts in both the environmental impact, the economics, if you can produce that much more, obviously productivity drives standards of living, and in terms of protein crisis, because you can produce so much more protein per land area. And so this just kind of summarizes those three areas that we're talking about. In the economy, you can look at it as the green revolution was a 2.3-fold improvement in productivity. We also brought in about 30% more arable land, and that meant for a 3-fold improvement in overall worldwide production of agricultural products for food. With algae, if you put 2 to 3% of the land that's currently going into terrestrial crops into algae, you could double that productivity. It's a 40-fold increase, and that is completely game-changing from an economic perspective. It's the kind of thing that can lift whole regions and areas out of poverty when you can produce that much more per acre. And it creates a full spectrum of rural jobs. On the protein supply side, we're already seeing over the last 20 years the big increase in cost from protein as protein has become more limited. All the major food companies were at a meeting in at the White House about 4 or 5 years ago. All of them were very concerned that by the late 20s, we would have severe shortages of protein. Algae solves that problem as well. And then, of course, in the environment, when you make better use of your resources, of your land, your water, and other things, that automatically ripples, well, should ripple right through to the environment and result in a lot of environmental benefits. So that was the basis on which we founded the company, was to try and figure out how do you make this happen. And so, in the words of another speaker from this morning, that was the good news. This is what algae could do. The actual news is the costs are 10 times too high for algae production to compete in those kind of commodities. And so that is a massive problem. That means every aspect of the process has to be addressed. If you have something that's 5% of your current costs and you bring it down 10 times, that's now 50% of your costs. And so that does require radical innovations throughout the process. And if I get nothing else comes of this talk other than I would encourage Berkeley to jump in on this. The founder of modern algae technology was from Berkeley. Professor Oswald developed the high-rate raceway ponds that are still in use in almost all commercial algae facilities today. Photosynthetic, again. I'm not speaking to heterotrophic algae. So the roots were in Berkeley. Steve Mayfield, one of the leaders in the field, is from Berkeley. John Benneman, one of Oswald's students, also still very active in the field. So if that's not enough Berkeley ties, I just want to encourage you. The last one is, I'm a chemical engineer. The problems are almost all in engineering. Well, not almost all. It's an interface of biology, physics, chemistry, and engineering. But there are tremendously interesting chemical engineering problems to solve. And so we need more chemical engineering schools that are of Berkeley's caliber working on these problems. That's my commercial. This is the basic process for those of you who are not familiar with it. Basically, we're taking CO2, water, and nutrients. We're growing just like you do with any plant. But we're cultivating those in open raceways, so open shallow raceways, typically less than 12 inches deep. We actually operate much shallower than that. We then produce an algae that's very dilute. It's 0.1%. So it's 1 gram per liter, and we have to harvest that and get it to a concentrated product. That's an important step. And then once you've harvested it and you have a nice slurry, then you need to both dry it and extract it. You have to dry it because one of the products is protein meal. That needs to have long-term stability and needs to be able to be used for food and feed applications. And then the extraction is to remove the lipids. The big benefit of algae, and one of the reasons why it's so productive over some of the other plants, is that you're primarily getting oil and protein and not that much sugar. So most of the cellulosic situations with trees and other things that are highly productive, you're producing primarily sugars, which are much lower value than protein or oil. And that's important because obviously algae is much more expensive to grow than those. This is the facility that we've used for almost all of our research and development. You can see we've got a series of open raceway ponds in the center. And then down on the lower left is a power plant. We're adjacent to a power plant because we get all of our carbon dioxide for growing the algae from that power plant. There's a pipeline that brings a slipstream from their flue gas over to an absorber on our site. Just to give you an idea of scale, that absorber is a 50-foot tall absorber in the lower right. The largest raceway is about 3.2 acres. And so even at the research level scales are actually not that small. But we need to get much larger, obviously, if we're going to be in commodities. So where we are right now is we're doing our scale up from that 8-acre facility, 8 wetted acres, up to 150 acres. And that's because we've now gotten all the major breakthroughs we think are needed in order to make algae economically competitive. And so if you look at this table, kind of has years, obviously, the breakthroughs. I know I'm not going to go through all these results. It's an eye chart, but the main thing is you'll notice almost all of them are at least 1 order of magnitude, if not 2 orders of magnitude improvement that was required in order to make it competitive. And the last chart I wanted to include, because you can see DOE is very prominent in there. The algae field would not be even close to where it is today without the DOE's funding through BETO. All the acronyms there just show that there are a number of different programs, even from BETO, just with our company, that were involved in leading to these breakthroughs. I'm going to go through a couple of those in a little more detail with some pictures, so it's a little more interesting than just a chart. One was scalability. If we're going to be able to produce a commodity, we need to be able to go to scale. That doesn't mean producing 1 million small reactors. It means we need to make one big reactor, right, and take advantage of scalability. We've now developed a system that we believe will be scalable up to 200 acres. We've gone through 3 orders of magnitude in scale now and been able to maintain our productivity and all of our hydrodynamic measurements consistent across all those scales. And so we're confident in that. We're going to the next level. The 150-acre facility will have about a 26-acre raceway in it. In harvesting, I put that up here just to give you an idea of this order of magnitude needed. We had a 2 orders of magnitude reduction in the energy use for harvesting. So it went down 100-fold. And on top of that, we also were able to get complete separation of the algae from the media, so we get a crystal clear return and produce a high concentrated stream of 15 to 20% slurry. Even all the current technologies couldn't meet any one of those other benefits, and they required much more energy. The next one is on extraction. Conventional extraction for algae takes about 2 megajoules per kilogram. It's usually hexane extracted. It's just a solvent extraction system, and then you separate the oil from the solvent. You end up producing a tar-like oil because the hexane pulls out a whole bunch of other stuff besides just triglycerides. We developed a new extraction process that uses a 20th of that energy, and yet produces a much cleaner stream as well, where we get a fairly clean triglyceride oil stream coming out. We're not pulling everything else out of the algae. And the last one up there is just on productivity. The lower lines on that chart are all kind of the baseline. DOE runs test bed facilities that generate baseline data. So that's the baseline data from those facilities. The blue line was our initial improvement in productivity, and then the green is where we are now, which is about a 2 to 3 times improvement in productivity. We've accomplished that improvement in productivity while reducing the energy that we use in those reactors by a factor of 10 as well. And so, again, it's not so much about the way we're doing, but understanding if you're in this field, you're looking at order of magnitude changes needed to make things economical. And this just gives a summary of those changes. So the yellow lines or the orange lines are conventional technology. DOE set out some ambitious goals. This is from a few years ago. Their goals are obviously much even tighter now, but they were the goals when we were starting. And we've met or achieved in all these particular metrics those goals, and in many cases actually far exceeded it. And again, just trying to emphasize the magnitude of the challenge here. This is the current technical economics based on our modeling. We're looking at getting down in the range of 50 cents a kilogram for production if we were at full commercial scale. That assumes, of course, everything that we're doing at 8 acres works when we go to 150 acres and works again when we go to 3,000 acres. And then the pilot plant design point is obviously higher because that's not a full-scale facility, but that's what we're working on right now. So, and just want to acknowledge DOE and DARPA both funded a lot of this work, and they don't. They're not guaranteeing any of the things I said were accurate, I think is pretty much the summary of that. So thank you very much.
Thank you, Dave. Our next speaker is Eric Mcafee. Eric is the chairman and CEO of Aemetis, and he will talk about commercializing below zero carbon advanced renewable fuels.
Thank you, and I want to start today by thanking Blake Simmons and Jay Kiesling and the other partners of ours at J-Bay. We're just wrapping up a $3 million California Energy Commission grant on ionic liquids that should have some real breakthrough opportunities in traditional corn ethanol, actually, by adding non-food wood waste as a feedstock for traditional corn ethanol plants. There's about $30 billion invested in 210 plants operating in the U.S. that comprise over 99.8% of current ethanol production, and it would be a revolution to bring a new feedstock to that industry that could be integrated with the existing facilities. So thank you for the team at J-Bay that has supported that. We also want to thank Beto and DOE for the leadership they've had in the biofuels industry. Our company is in the business of commercializing advanced biofuels. We are a NASDAQ-listed company, so there will be tests on this slide when we're done. If you wouldn't mind, the lawyers would appreciate me to highlight that. Our mission, though, is to reduce global dependence on petroleum. And the name of our company comes from A, which in Scottish means one, and Greek, Metis is the mother of the goddess of wisdom, Athena. So in Greek, it's prudent wisdom. So the name of our company is One Prudent Wisdom, which we applied to reducing our dependence on petroleum in our generation. And I say it in our generation because if we don't, that means our kids are going to be fighting wars in the Middle East and fighting over geopolitical power based on petroleum. And our generation has an opportunity and a responsibility to undertake technology development that releases us from that ongoing commitment to lives lost and money wasted. First generation biofuels is primarily driven by corn. Why? Because we all like to make beer. And what you do in a corn ethanol plant is you make vodka, a really good quality kind of beer. And there's a tremendous, really low threshold on producing the product. It's been a tremendous focus on yields. Second generation, where we actually imported 60 million tons of Milo from Argentina, we demonstrated, and this was part of a California Energy Commission grant process. And then third generation is taking waste, primarily woody biomass. There's about 1.2 billion tons of it available in the U.S., according to Beto's annual report, and turning that into biofuels. We'll talk a little bit about what kind of biofuels, but in general, the market today is primarily ethanol, about 15 billion gallons in the U.S., and biodiesel, about 2 billion gallons in the U.S., and migrating toward renewable diesel as a product. But all biofuels start with one question, and it's not the technology of conversion. The question is, what feedstock do you have? And today, the feedstock is corn. Why? Because the technology has not been commercialized to convert anything other than corn into a biofuel that's valuable from a biomass. So, overview, we have 4 lines of business. We own and operate a 65 million gallon capacity ethanol plant in California. It's the largest ethanol plant and frankly, biofuels plant in the state of California. It's out in Modesto, about 50 or 60 miles to the east of us. We also have a 50 million gallon plant we built in India. Many people have accused us of having a strategy drift, but interesting enough, that was the first plant I built. And it's because I'd founded a company called Pacific Ethanol and took it public. It became worth almost $2 billion in 2006. I sold out my founder's stake. And I did not want to be correlated with the US federal regulatory policy regime. So I found that the farthest away you can find from the US regulatory regime is the country of India. And we built a 50 million gallon plant, which, by the way, has only taken 14 years to become nicely profitable and successful and rapidly growing. So if you have an extra 14 years to invest, India is a great place to go. It has panned out beautifully for us, but it literally took 14 years to get it done. We're building a 12-dairy biogas digester. It's the first quarter of what eventually will be about a 50-dairy collection system. This feeds biogas to our ethanol plant and also into the PG&E pipeline as we start injecting later on this year. And then we have about a $200+ million cellulosic ethanol plant. We'll talk about it a little bit today. But this is a revolution in what feedstock is available for ethanol production. I want to highlight for everyone here, I think we're primarily talking scientists and program managers and the like, and we all know this. But in any slide deck you're reading, please start with the management team and the board. On our board for more than 12 years is the former Secretary of Agriculture under Ronald Reagan, Jack Block. On our board is the first woman corporate officer of Chevron Corporation. She spent 38 years there, but her last 20 years was secretary. She attended every single board meeting of Chevron Corporation for her last 20 years. She joined our company as one of only 2 companies in her retirement. The maturity of the board, the maturity of the management team, this team's been over 12 years has been together, is very important. Because I'm sitting with scientists, I always like to show this slide. It usually results in tremendous conflict and irritation. Please don't do that to me today. This slide is a statement of why we like biofuels. Number one, by the way, I founded a $1.3 billion solar company, so I'm a solar guy as well. I also founded 4 oil companies that took public, so I'm an oil guy. So you can accuse me of being on both sides of all these issues. But here's a statement of a type of reality I'd like us all join into. Solar, wind, hydro, and nuclear electricity are tremendous. We're avoiding using other types of petroleum or coal, carbon out of the ground, right? We all bought into that. We all agree with that. The one thing it can't do is actually consume carbon dioxide from the atmosphere. In order to do that, you have to have some absorption mechanism. Nature calls them trees or vegetation of some type. They grow by sucking CO2 out of the atmosphere along with H2O. They make biomass. So renewable fuels and biogas are the ways that we can actually not just reduce how much petroleum we use in the future, but actually reverse climate change, reverse the amount of CO2 we have in the atmosphere, frankly, dramatically reduce air pollution as well. And if you're not committed to biofuels or to renewable natural gas because you have a religious commitment to electricity, think about this. How do larger electric cars actually operate? They need a dense energy source on board. What if that's a negative carbon emission, renewable energy source, domestically produced, creating jobs and investment in the US to run your electric car? Wouldn't that be a lot better than coal and natural gas, which comprise about 60% of our current electricity grid? Wouldn't it be more intellectually honest to say if we're going to take the equivalent number of joules of 10 times our current grid, which is the amount of BTUs required to run our transportation system, and dump it on the electricity grid, that we're going to actually be more dependent on coal and more dependent on natural gas when we all get committed to electric vehicles as the only solution? I'm saying this in the hotbed of EVs. This is CARB country. And we all are agreed we're never going to have an ICE engine, an internal combustion engine in the future. But let's get serious from a scientific point of view about understanding how electric vehicles will be powered. Where's that electricity going to come from? And what you'll find is that Nissan Motor Company, the largest motor company in the world producing electric cars, said that's going to come from the ethanol molecule, reformed on board the vehicle, either through an internal combustion engine or through a fuel cell with no moving parts to actually power your electric vehicle. That will give you tremendous efficiency in your electric motors, all the performance of no sound, all that kind of wonderful stuff. But make you not have to plug into a coal and coal and natural gas grid to power your supposedly clean electric vehicle. This lack of scientific rigor is damaging to our children and our children's children as we create bad policies, as the speaker just before us talked, and those bad policies then get promulgated in commercialization. Sorry for hitting you up with that slide, but please talk to me afterward if you want to argue about it. California, about 80%, 87% of all the carbon reduction in California has come from electric cars? No, biofuels. Electric cars aren't even on the slide. So if you're really committed to carbon reduction in California, you're talking about ethanol, renewable diesel, biodiesel, and biomethane. That's where you get carbon reduction. You can power electric cars from those sources, but if you're not starting with those sources, you're not being scientifically rigorous about carbon reduction. Now, if you look at it, I mentioned our companies in commercialization. If you look at it, you want to start with, well, what are the economic values if I go and I create those carbon credits? And you'll notice down the bottom that on the far left, the vertical line, by the way, is zero carbon. So on the far left is biomethane. It's in the negative 270 to negative 350 negative carbon intensity. So the number one most economic opportunity you have is to actually capture biomethane, don't emit it into the sky as a 34 to 100 times worse carbon greenhouse gas emission than CO2. Instead, capture it and either destroy it or use it for something productive. And so we are investing about $50 million this year in capturing the methane from about a dozen dairies. There's 1,250 dairies in California. We supply about 100 of them with animal feed every day, 50 trucks, feeding 100 dairies. We've gone to those dairies and said, guys, let's put in very substantial investment, capture your biomethane, and you're now on this slide as the lowest carbon by far of any potential fuel source. What this does is displaces diesel in heavy over-the-road trucks. And so our system, which includes building a pipeline and building digesters and building gas cleanup at the dairy and also at the head end and then interconnection, and it's $50 million I'm spending, is with one simple goal. To answer the obvious question, who's going to step up and start capturing the biomethane, which is the most economic and, quite frankly, because it's so damaging, most productive fuel source for California. The second thing is the little dot you see there near the top where it says filed. Cellulosic ethanol is the second most negative carbon source you have in California. Taking waste wood from orchards and the like is really the second way to do it. This slide is for anybody that wants to afterwards ask me why I talk about ethanol so much. It's 114 octane. It's racing fuel. When I was a kid, I'd love to have that in the car that I was driving to high school. It's 34% oxygen. It's an oxygenate. It replaced MTBE. It's the stuff that makes gasoline burn cleaner. It's the campfire in your engine that now has more oxygen in it so it doesn't put out as much pollution. We all like that. I especially like it. I grew up as a farm kid near Fresno, California, which is now the second most polluted region in the entire United States, according to the EPA. It's also the fuel source that Nissan Motor Company selected as a renewable fuel for the future fuel cell to drive their electric vehicles. It is a fuel you can put into an internal combustion engine and make that electric car carbon negative by generating the power on site in the vehicle. And so that's actually a very important part of our goal, is to create the molecule that can be used by the electric vehicle industry to actually do heavier duty vehicles. So as I mentioned early on, biofuels is all about feedstock. And in the Central Valley of California, we used to take the orchard wood from about 1.5 million acres of almonds. And every year there's about 3.2 billion pounds of waste wood that comes from those almond orchards being knocked down after they have 20 years of productivity. And you might notice that on the lower left graph there, we were using that to make electricity until the federal government passed the solar and wind support of a 30% investment tax credit. And then we stopped doing that because it wasn't economic anymore. So Central Valley of California now is 1.5 million acres of almond and walnut orchards. You chop them down every 20 years, and you create what? Air pollution and carbon emissions. We are probably the nation's #1 best producer of air pollution and carbon emissions by burning orchards in the Central Valley of California. We turned that into feedstock. We went to UC Davis, said, hey, give us a study. What's the future going to look like? And they said the future is going to look worse. Top left, we know what the next 20 years worth of orchards look like, orchard waste would, because they're already planted. They were planted this year and for the past 20 years. And so they said the feedstock's going to become even more available. And so we put together a business to go and invest in converting this waste wood into low-carbon ethanol. On the stage today is one of the world's great scientists and inventors in biofuels. Books will be written about him, statues will be built in his name, songs will be sung by young children. He gets to talk next. But clearly, this young man from New Zealand came up with a process that we are investing not $250 million, but probably $2 billion in to solve that problem I mentioned in Central California of air pollution and carbon emissions, and turned it into the fuel for both gasoline engines as well as electric engines, electric vehicles. And that is an ability in the top, what is it for you? Top right, to take a gas, ferment it with a microbe that this scientist discovered and spent his life commercializing. And that microbe being fed gas, instead of yeast being fed sugar, allowed a revolution in feedstock to happen, because we were able to gasify the feedstock that feeds this microbe. So, thank you. So we went through a process of commercialization, which included building a demonstration plant for the U.S. Department of Ag. We went and found a site. It's out near our existing corn ethanol plant. It's a 140-acre former Army ammunition plant that we're turning into an advanced biofuels facility. And we went to Tom Vilsack, he's there in the middle. He's the USDA secretary, and went through a long, expensive process and got a $125 million loan guarantee, and had to meet all these milestones to be able to do that. The federal government was rather rigorous about this entire process. And then we laid out a process to build 4 plants in the state of California, and then eventually be able to migrate across the country as we solve the billion-ton per year feedstock availability in the U.S. So I invite you to come and visit our website, ametis.com, learn about what we're doing, but probably equally as important, get to know our people. With us today is Jeff Welch. He's our Vice President of Strategic Projects. He's well known to many of you. If you don't know Jeff, please find him and say hello. And we offer ourselves as a potential partner to many of you as you try to work on developing your processes. We do have a breadth of business that's kind of unusual. So I thank you for your time today. Look forward to talking to you later.
Thanks, Eric. Our final speaker is Sean Simpson. Sean is co-founder and the Chief Scientific Officer at LanzaTech. And the title of his talk today is The Pollution Solution at Scale, unless it's changed. No.
Cheers. Thanks for that. Worrying. I'll press this here. Press it again, press it again. So as we've heard sort of repeatedly today, the manufacture of sustainable bioproducts is primarily today based on the conversion of sugars, so sugars from corn in the US, sugars from sugarcane in Brazil, into a variety of things such as ethanol. Ethanol is produced today between North and South America at a rate of around 23 billion gallons a year. But the challenge we face is to produce at many orders of magnitude greater than that. The challenge we face is to displace as large a portion of the 90 million barrels a day of oil that the world consumes with equivalent products. And for that, I would argue we have a feedstock challenge. Wouldn't it be great if we could use this stuff to make sustainable products, which today we're sort of cleverly using to feed fish? Wouldn't it be great if we could use this, industrial waste, above ground waste carbon resources to produce sustainable products? Or the waste products of agriculture, any agriculture, regardless of what form that biomass looked like. And effectively, that would allow us to produce sustainable products anywhere in the world, not just in the agriculturally rich areas of the world, but anywhere in the world, and therefore achieve a scale of biomanufacturing that today we can only really dream of. And in future, wouldn't it be great if we could actually use abundant and extremely low-cost electrons to fix carbon out of the sky? At LanzaTech, we focused on gas fermentation. And we focused on gas fermentation because we wanted to identify a technology that would allow biomanufacturing at enormous scale, anywhere in the world, to produce sustainable products at impactful volumes. Gas fermentation allows each of these resources, waste from industry, waste from society, and waste from agriculture, to all be converted into products using a microbial catalyst that accepts gases, carbon monoxide, hydrogen, and carbon dioxide as the sole source of carbon and energy for the synthesis of both fuels and chemicals. The waste resources we can access are pretty much available everywhere. And going forward, we see an opportunity to leverage electrons as the source of reducing equivalents to fix carbon dioxide into these products. We've, at LanzaTech, have developed and now scaled and commercialized a process to produce low-carbon fuels from each of these resources at scale. We developed the microbes, and we've developed the fermentation processes from end to end to achieve this. At first, we're focusing on the production of ethanol, but we've also developed synthetic biology techniques to modify our microbes such that the carbon and energy they consume are redirected into new products. This is a picture of our first commercial plant. This is operating in China. This plant produces 16 million gallons a year of fuel. It takes a waste emission from a steel mill and processes that directly into a low-carbon fuel. To date, we've produced over 11 million gallons of ethanol at this facility. This is a continuous technology. We continuously feed waste gases in, and we continuously produce ethanol. The microbial catalyst that we used is also continuously produced, and as part of the process is separated and actually sold as an animal feed product. This technology, this is the first time this technology is being commercialized at scale. We commissioned the plant in late 2018, and it's been operating ever since. The process is profitable. We have between a 3 and 5 year payback period on the plant. We pay for the gas going in. There's nothing for nothing in this business, as everybody knows. We pay for the gas going in because the opportunity cost here reflects the BTU content of that gas. And the partners who have commissioned this make a profit. We're now in construction with a similar plant in Europe, again at a steel mill facility, converting an off gas from a steel mill in Ghent with ArcelorMittal, the world's largest steel maker, as our partner. We're in construction of a plant in India with Indian Oil Corporation, taking a waste gas from one of their refining units in a refinery above Delhi in a place called Panipat. We've currently in final design for a facility in South Africa, converting a waste gas from a ferroalloy plant. In Japan, with a syngas produced from municipal solid waste. And as you've heard in California, with a plant using syngas created from local orchard waste. Within 2 years, we anticipate commercializing our process in the context of each of those major waste streams that I identified earlier, in order to demonstrate to the world that we can think about our above-ground carbon resources in a very, very different way in order to produce very large volumes of fuels and chemicals to genuinely impact the need to consume 90 million barrels of oil a day. The volumes speak for themselves. The steel industry alone represents a waste gas resource that could be used today to produce over 50 billion gallons of ethanol a year. Municipal solid waste globally could be used to produce an equivalent amount of ethanol that is today produced from corn and sugarcane. Refining and agricultural waste is almost an infinite volume. And these are all processes that allow us to produce fuels in a very localized manner, distributed production. But who wants all this ethanol? is one of the challenges that we faced. And so one of the projects that we embarked on within the company was actually to look for alternative uses for ethanol. Ethanol is a great platform molecule. And so in a quite successful partnership with a government lab, in this case Pacific Northwest National Labs, and using very generous funding from the DOE and DARPA, we were able to take a catalytic process initially demonstrated in Pacific Northwestern National Labs, and we were able to take that catalytic process and scale it to show that we could turn ethanol into jet fuel and diesel. In fact, the process was so successful that we were able to get a pilot plant operating in a relatively short space of time, produce several thousand gallons of jet fuel that we used then to gain certification within the ASTM to allow us to sell this jet fuel in the commercial aviation industry. And last year, we put some fuel into this guy's plane and glamorously flew from Orlando to the UK. Our plan is to commercialize this technology within the next year, and we're going to spin out a separate vehicle in which to do that, which I think we're going to imaginatively name LanzaJet. This fuel not only is a sustainable aviation fuel, but it also allows a reduction in contrail production because the efficiency of burn of that jet fuel is so much higher. So fewer soot particles and fewer contrails production, which again is a greenhouse gas contributor from the aviation industry. As I mentioned earlier, we've developed synthetic biology techniques to allow us to modify now very precisely and very reproducibly the pathways within our proprietary organism to allow us to produce molecules that can be used in the production of acrylic acid, in rubbers and packaging and building materials and so on and so forth. We've produced actually over 100 different molecules to date, and we're focused on two specific molecules that we intend to commercialize towards the end of this year, namely acetone and isopropanol. And the reason to focus on those initially is that we want to allow the plants that are today producing ethanol to become flexible such that they can in future produce chemicals with the same hardware that is today being used to produce that ethanol. In this way, again, people have talked about this, we're talking about sort of hardware, software models where microbes are being switched out of plants in order to allow the production of multiple different products at using the same fundamental hardware and with minor changes to downstream processing using molecules that effectively play well together. So really what LanzaTech is trying to become is this kind of missing link in a circular economic model whereby above ground carbon resources can be recycled into fuels and chemicals, or above ground carbon can be captured and reused into everyday materials. And we're hoping that within the next 2 years, that will happen in a very material and a very commercial way. Thank you.
Thanks, everyone. I'll start with a question or two, and then we'll move to questions from the audience. For your individual companies or in the field that you're in, what do you think the most pressing challenge is to scaling up, or in the case if you already feel you're scaled up, for really scaling out and being, in every state, for example? Maybe we'll start with Dave, since.
So we're doing design of a scale-up now. I mean, obviously, funding is obviously one of the key factors in any scale-up, and so we're working right now, developing those partnerships to be able to obtain the funding. I think from the standpoint, from a technical challenge, everything we've done has been about focus only on solutions that we think will scale well. And so we've put a lot of effort into seeing how they scale over the range we have. And so we're actually pretty confident that they will scale well. But part of that also means designing the facility so that, and our funding such that we have time to solve the issues that you don't expect that come up when you move to the next scale.
I think investors and management teams are struggling to find out what government policy is going to be 5, 10, 15 years from now when we're building facilities that will be operating for 30 years. And so enforcement of federal law, primarily through the EPA, is the biggest challenge we have, because it's very difficult to sit down with an investor in Connecticut or New York and say, here's what today's policy is, because if you read it, the federal law is not being enforced. So when the basic investment thesis is risk factor number 1, federal law will not be enforced, that kind of undermines your credibility as a management team and as an attempt to make investments. So anybody knows somebody at the EPA, please call them and tell them to read federal law and then enforce it. It'd help us a lot.
Sean?
Yeah, I mean, I think for us, when you're scaling a brand new process, you naively think that the technical piece is going to be the challenge. What we found actually is policy is probably the greatest challenge. And that's because we're offering an opportunity to use brand new feedstocks that current legislation simply don't support the use of. There is no support in the US for the use of a waste emission from a steel mill today. If I make ethanol from a steel mill waste gas in the US, I cannot sell that ethanol under the regulations within the RFS. Not supported. Low carbon, yes. Provides industrial jobs, yes, but not supported, because the renewable fuel standard specifically sort of encourages the use of fuels made from biological material and doesn't recognize the fact that we need to address our blatant dumping of carbon into the atmosphere. And so that's something I think that is a major barrier to innovation in this country, because what we've done is basically put in place policy that says thou shall convert this stuff into things that we want, and not said we will reduce carbon emissions and bring forward your ideas, and that's unfortunate.
Yeah, interesting. Trying to have the policy or legislation really track with the new innovation in a way that really fosters economic growth.
Well, it's not, I mean, it's just not technology agnostic, it's not feedstock agnostic, and it's not focused on an outcome.
Are there any questions from the audience?
Seth Snyder from INL. I'll start with Sean, but it could really go to anybody. So how come you're succeeding when Coscata and Ineos, they're off in the sunset? And all of you, there's so many algae companies that sunsetted, in the conversion companies. There's so many failures, but you're succeeding. And that's, I think, an important message today. I'd like to hear what you think.
Why?
Why?
I think, it's difficult. I don't know. It's difficult to say why someone else didn't succeed. What I can say is that, we're very bloody minded. So, and we also made the call actually to commercialize in China first. And we were able to get partners in China that did take a risk on technology, huge risks on technology, and were willing to consider the use of feedstocks like a waste from a steel mill is the first place to go. And they were willing to invest in a first-of-a-kind process to achieve this at enormous scale and cost, and took a real risk, and it paid off. So we were really the beneficiaries of a kind of future-focused, risk-accepting attitude within China.
And I just say, from an algae perspective, I think one of the biggest problems has been not recognizing the magnitude of the problem, which is why I tried to emphasize that in the talk. That most of the companies started up, they had very good ideas on how to solve one of the issues or one of the problems in the process, but didn't recognize all that was needed in order to make that work. And so they felt they could scale up conventional technology with one minor, not minor, but even a major improvement in one area, and then that would be economical, and they just didn't see that. I think part of the reason for that is they were blinded by the tremendous benefits that I started with, right? The good news is so good, people ignored some of the news that they had in order to get there. And then I think a part of it is exactly what you said. We've been stubborn about it. We said, look, we're not going to pivot to high-value product. We're not going to stop on any problem until we've solved it. And so I think that was a factor. And then I think we were also had the benefit that I had the fortune to lead a group of about 30 companies trying to develop algae technologies for DARPA in the around 2000, over 3 years really, from 2009 to 2011. That gave me exposure to really all the problems. So when I started this company, I didn't have any misconceptions about how big the challenge was. And I knew we had to have solutions to all those before we scaled. Now, I would argue we're not successful yet. We're still pre-revenue, right? We're scaling now, but I think we have definitely shown success in terms of solving some of these major problems that have been out there for a long time in the industry.
This is an unusual panel because on this panel are the three most suffering people from each one of our respective companies. It is sad but true that unless you have a long-suffering, charismatic, visionary leader, it's very, very hard to survive the mortgage crisis and then the oil price crash of 2014 and 1,000 other things that would kill off a regular company. And it helps to have a management team and then a board of directors and then investors that then kind of get into this reality distortion trap that you create for them. So they all really believe this is going to happen, despite the reality that there's so many factors against you. And I think what you're hearing from many of us, though, is a focus on commercialization. The science is very interesting and very compelling, but what we get excited about is we can actually create a product out of it. We can get it to market. We can build things, and that's what brings the broader group together. What I find, I'm a venture capitalist, by the way. I funded about 25 companies, is you have a scientific founder who's not that interested in commercialization. It's an irritation. And to have a scientific founder that's very interested in getting their technology and their product to market, and that's why they are in the business, is something very special. And as you're looking to give grants or to support companies, it's sad to say, oftentimes the best thing for a scientist is to find the rest of the team, and then you fund the team. The team's going to create the value. That one visionary leader oftentimes doesn't have the depth.
I add one other thing that I think that has been helpful. It's actually, Moran answered a question earlier, what happens when you have 1,000 ideas and how you narrow it down to what you do, because we certainly have over 100 ideas on our development map right now. And I think what we've done that is, one thing that's helped us is anytime we have a new thing, we say, what is the fastest way you can prove to me that won't work? That has two things. One, you don't waste a lot of resources on it, but most importantly, it eliminates confirmation bias, which is what sends you down a lot of rabbit holes for a long time, because it's easy to see all the things why this might work, but if you're really focused on trying to prove that it won't, then you can find the ones that are good a little bit faster.
So we're out of time. Could everyone please thank our panel again?
That was awesome.
So it was a great panel. Thank you. I'm back up here to introduce Chris Fall, who's director of the DOE's Office of Science. I've been working with the Office of Science for many years, and actually it's attracted some really great people to that job, which is great for the country, and Chris is another one of those. I remind you that the Office of Science is the lead federal agency supporting fundamental research for energy and the nation's largest supporter of basic research in the physical sciences and engineering, I believe. He oversees the two principal thrusts of the office: direct support for scientific research and development and construction and operation of unique access scientific user facilities. So there's both this grand research enterprise and these facilities that are unique in the country. The Office of Science is also responsible for stewardship of 10 of the 17 national labs. Berkeley Lab is one of those 10. And before this role, he served as a senior advisor to the Undersecretary of Energy. He was acting director of RPE. He served various roles in ONR, in the Office of Naval Research, including acting chief scientist and lead for the Research Directorate and deputy director of research. And then on loan from ONR, he spent some time at OSTP, the Office of Science and Technology Policy, as assistant director for defense programs, and then as acting lead for the National Security and International Affairs Division. So he's had a very long career and a very effective career in government service, and we're pleased to have him now as the head of the Office of Science. So please welcome Chris Fall.
Thanks, Mike. I got the subtle message. Directors of the Office of Science come and go. And it's true, right? It is actually true. And I just let me throw it back at you that we have a number of the laboratory directors here today. I'm interacting with them during the networking sessions. These are just remarkable people. If you think about the jobs that they have, managing the laboratory through wildfires and all sorts of other things, worrying about the budget, doing good science, which is the first mission, and then dealing with the political folks that swing in and out and have different sorts of agendas. It is a very rarefied group of people that run these laboratories, and we're grateful. I think the country's grateful to have people like Mike and the other directors here. So anyway, yes. Good afternoon. Thanks for having me here. I think it's been a pretty good meeting, but I want to spend a few minutes. We had the panel sessions; those were great. Started some topical discussions. I want to focus back a little bit on the laboratories, a little bit on the Department of Energy, and talk a little bit about the opportunities we have for innovation and in relationship with you all in the private sector and academia in particular. So thanks for being here. Thanks for staying here. I think I am the last person before Mary wraps up. So I'll keep it short. It's a lot of paper here, but it's a very big font, so I'm getting old. Anyway, one of the great things about these XLab meetings, and this really is an innovation, so hats off to Connor, hats off to the Undersecretary for thinking about these things, is the opportunity to think about this idea of the pipeline from basic science to transition, and the role of organizations like the the Department of Energy, Office of Science, and your organizations. How does that all fit together? The Department of Energy has an incredibly broad research portfolio, right? On the one hand, there's the Office of Science, basic science. I'm going to talk a little bit about that, most of my remarks. We have the applied. You heard a little bit in the panel sessions about the applied offices, the Bioenergy Technology Office in particular in the energy efficiency and renewable energy applied piece of the Department of Energy. Daniel Simmons, you'll hear from him tomorrow. I think he's here, the Assistant Secretary for Energy Efficiency, Renewable Energy. We have Connor in the Office of Technology Transition, another piece of the innovation puzzle. And so this basic applied tech transition, all the pieces are there. So how are we going to apply them to the bioeconomy? What I want to do is focus on the Office of Science. That's my responsibility. And tell you how we think the Office of Science fits in to the bioeconomy. So in 2020, this year, fiscal year, record budget, as the Undersecretary said this morning, $7 billion. That's quite a lot of money. Our mission statement is to be the nation's largest funder of the basic physical sciences. So we have particle physics, nuclear physics, fusion energy sciences, broad spectrum energy sciences, biological and environmental research. I think Charlene Weatherwax is sitting over here in the audience. And as a supporter of this basic research portfolio, we have two main responsibilities. First, and most importantly, is to be good stewards of basic discovery science. That's a mission we don't, for all the importance of doing translational work, transitional work, developing the economy, growing jobs, our real job is making sure we have this basic science base and being good stewards there. But the second is to ensure that our society, our people, our economy, and in some sense, people around the world reap the benefits of that basic research in the form of innovation whenever possible. So it requires that we be alert to the possibilities, to the basic science discoveries that could lead to innovation, that could lead to jobs, to growing companies, and it also requires an informed relationship with folks outside of our laboratories and outside of our programs, and that's why we're here today. That's why we've engineered this now 18-month-long XLab series. It's to connect with you all outside of our, look outward instead of inward. So I did say that the Office of Science has the basic physical sciences as its principal mission, but we're here to talk about the bioeconomy. As far as I'm aware, there's no agency in the federal government whose job it is to be the executive agent for the bioeconomy, right? I don't think there's an agency in the federal government who has the executive responsibility for biotechnology, right? We don't have that. We don't have that yet. Could be a possibility for the future. But the Department of Energy, the Office of Science in particular, does have a large biosciences footprint. And I think that we have some superpowers, if you will, that we can apply to growing the bioeconomy. So as a case in point, I think the biotech revolution over the past 3 decades provides one of the most, across any areas of technology, one of the most compelling illustrations of the opportunities at the intersection of discovery research and innovation. And it's a story that the Department of Energy played a key role in, right? I think you know what I'm going to talk about. Maybe I'll keep you waiting for a second. You've heard about it before. Anyway, key to the role is science at scale. So science at scale is one of these superpowers that we talk about that the Department of Energy has and the Office of Science in particular has. So from our own laboratories and our incredible user facilities, we talked a little, the Undersecretary talked this morning about user facilities like our X-ray light sources, our neutron imaging centers, to the massive international collaborations like the Large Hadron Collider at CERN. That's done by the Department of Energy Office of Science. We know how to do big science. We know how to go big, right? And back in 1986, we kick-started, the basic science was there, but we jump-started, I think, the biotech revolution sort of into high gear by doing just that, by figuring out how to scale and go big. And that was the year when the precursor to the Office of Science proposed something pretty novel and revolutionary, and that was to sequence the entire human genome, right? I don't think anyone in 1986 knew whether that was possible. It's very fashionable these days. We go to a lot of meetings to talk about moonshots, right? Everything's a moonshot. Well, that was a moonshot idea, right? A pretty, big risk. Not sure if we can get there. Obvious, the scale of the investment at the time was going to be big. And we decided to go forward. And from that idea came the wider Human Genome Project. 3 of our laboratories, Berkeley, Livermore and Los Alamos partnered along with the National Institutes of Health to make that happen. And you guys know most of the rest of the story there, but then, once we use that problem, that moonshot problem, as an organizational center for these laboratories, then evolved the Joint Genome Institute, these labs coming together, bringing their expertise together, their different perspectives together to create something new, and that I think has just now been relocated to the Berkeley campus in the new building. So in turn, the Human Genome Project spurred the creation of all sorts of new technologies, genomics at scale, that have dramatically improved health outcomes, lots of opportunities there, and given an engine for innovation for the broader biotech revolution. So $3.8 billion was spent on the Human Genome Project. I think that's the right number. What was the outcome? In 2011, Battelle estimated in a report that between '88 and 2010, the Human Genome Project generated nearly $800 billion in economic activity and created more than 300,000 jobs. These are difficult things to estimate, and of course we pay Battelle a lot of money to manage our labs, so it's okay, but whatever. What's the number? There's a huge multiplier effect here, right? 3.8 billion to some tens of multiples of that just in economic activity, and then all the jobs that go with it. 3 billion base pairs in the human genome. It took multiple teams more than a decade to do that. And I don't know if this is the right number because it seems incredible to me, but the number I was given is now the JGI sequences about 300 human genome equivalents per day. Is that right? I don't want to put anyone on the spot, but okay, sounds good. 300, big. That's science at scale. That is science at scale, right? Gone from a collection of people in a cottage industry, put them all together, applied this superpower of the Department of Energy, and now we do science at scale in a very big way. So another superpower that the Department of Energy and the Office of Science has is that we're not limited to funding individuals or virtually collected sort of groups of investigators, right? We own these laboratories. They're operated for us by our partners, but we own the laboratories, and so we can build physical infrastructure that lets people to come together, share resources, and collaborate in new ways, right? One case in point that the Undersecretary mentioned this morning is the Bioenergy research centers that have been formed in the DOE. This grew out of a report from the National Research Council, so another important aspect germane to why we are all here today is that it's a big tradition in the Office of Science and the Department of Energy and our research programs to collect input and develop our directions based on the scientific community. So National Research Council made some recommendations, and so we developed this new organizational structure for research and established the 3 multi-institutional, multidisciplinary centers originally focused on cellulosic biofuels. That mission has expanded over the years. And these centers, Lawrence Berkeley, Oak Ridge, University of Wisconsin, and Michigan State have one together, have been pretty successful. We just added a 4th at the University of Illinois. The Undersecretary this morning gave you a lot of statistics about the intellectual property that's come out of those organizations. It's less obvious, but completely critical when we think about bringing people together, academia, industry, and so forth, that these organizations serve as a pipeline for training talent, right? There's a STEM education component to these, a technical training component that comes along with these centers. That's critical. And they also serve as organizing centers for wider coordination and collaboration. So they have significant relationships with industry, they have partnerships with private companies, and then industry advisory boards. So all the sorts of structure that goes along with something like that for interaction also comes out of these kinds of new ways of working together. Let me foot stomp again the user facilities that we support on behalf of the country, and say a little bit about what they can do in the context of the bioeconomy. The user facilities, I think we have 27 of them. These major facilities are all accessible for open science. It's peer-reviewed. We support that work. If you're peer-reviewed and you're selected, wherever you are, international, domestic, that's on us to pay for. And of course, there's absolutely access to the private sector. You have to pay the freight, or most of it anyway. I don't think it's quite full cost recovery. We'll say it's full cost recovery. But obviously these facilities are open to the private sector as well. We mentioned JGI, that's a user facility, the Genome Sequencing Center. We have the Environmental Molecular Sciences Laboratory at PNNL, 5 nanoscale science research centers, 5 X-ray light sources, 2 neutron facilities, with plans to build another target station at our spallation neutron source. Oak Ridge is represented here. Thomas, I'm sure, is in the room. There he is. And that second target station is really optimized in particular for at energies that are relevant to the biosciences, so that's useful. Another of our superpowers, another of the superpowers of the Department of Energy, is the ability to work in what we call convergence, right? Convergence in the biological sciences, the combination of biology, mathematics, the physical sciences, and cutting-edge computation. Nowhere else but at the Department of Energy laboratories does this kind of thing come together at scale. You know, bringing all of this expertise together, and oh, by the way, with an X-ray light source or a neutron source or one of the world's most powerful supercomputers, or at some laboratories, all of these things together, right? Nowhere else does that happen. And oh, by the way, at laboratories where the best minds in the world, not just from the US, want to come together and collaborate. This is profound. It's really profound, and it's crazy. It's on Connor. I'm looking at Connor. One of Connor's other jobs, many impossible jobs, is to tell the story of the labs and make sure that everyone knows all the great things that we do. Small job, you can do it. Very capable guy. Anyway, it's profound. So science at scale, right? The convergence of disciplines, physical places with unique collaboration models and focus goals, the appetite to take calculated risk. These are the sort of superpowers that the DOE and the Office of Science provide to the country, and we think that these are applicable to the bioeconomy as well. So we really do live in a golden age. I'm sure the Undersecretary said that this morning, of scientific and technological achievement, and not least for the biological sciences. We have a lot of you've heard about some technologies here. I do want to just pause for a second and remind everybody that technology can be a double-edged sword, right? So we understand that the synthetic biology tools and approaches needed to supercharge, to realize the possibilities in the bioeconomy have to be developed and deployed in, secure, predictable ways, so that on the one hand, we know that the results are going to be consistent and safe. There's the health aspect, and there's also a national security aspect. So I'm really happy. I think it's tomorrow that we're going to have a session on ethics, yes, but also the security of modern biotechnology. That's something we're focused on, having a robust biosecurity base, having the talent, having facilities within our national laboratory complex is one of my priorities. I think that, we're moving out to figure out what this is going to look like. You'll probably hear more about that in the future, but we need to have folks that understand this and that can straddle the line between opportunity and security within the complex. Just open the paper and look at what's happening today over in China. So there you go. So my main goal today was to tell you what we have to offer and what we think that we can do for you. But I'm going to say I'm really proud to be part of a leadership team at the Department of Energy level and also in an organization, the Office of Science, with a tradition that embodies, that embraces the premise that we don't have all the answers, right? Really, this is a philosophy of this leadership team, a tradition of the Department of Energy Office of Science, that we need input from the community. I said that starting out as we chart our course in the various things that we do. So help us, right? We need your ideas to help us shape the collaborations, decide on areas of emphasis, to contemplate new user facilities if we need those, dedicated to bioeconomy, biotechnology, and so forth. As I said, there is no other agency whose job it is to do this right now. That could come. We don't have that, right? And so here's the opportunity for you all at this convening, at other convenings, to tell us what you think you need from us, right? We're not, it's not appropriate for us to do this in a top-down way, and of course it obviously would not be successful. So, and just sort of closing out here, many of our laboratories have considerable strength in biotechnology. I'm not going to go through the list because I'm going to forget one, and then I'm going to catch grief at the lab directors meeting later on. But we are looking to make investments, right? We're looking to expand the investments, the relationships. We want to supercharge this biotechnology capability in regions around our labs, in Boston, in the Pacific Northwest, in San Diego. There are lots of places where there's already a culture of biotechnology. We want to figure out how to plug into that and do better. But I do want to say that we're in the Bay Area here for a reason. We're having the XLab in the Bay Area because of the special convergence that it offers, right? You've got 4 of our laboratories. You've got a robust biosciences base, several amazing universities, Berkeley, one of them, an entrepreneurial energy and entrepreneurial tradition, and then the financing, venture capital sorts of stuff that can leverage all of that. So I've asked our 4 Bay Area laboratory directors, not just what can the Office of Science, what check can we write, for next generation biology or bioconvergence, biosecurity, the bioeconomy, whatnot, but I'm asking them to think about coming together, right? We have these 4 laboratories here. They actually have relatively complementary strengths in biotechnology, and think about whether we can come up with some sort of intermediate step. We don't have a national biotechnology laboratory, but we can think about a sort of virtual construct, work together and come up with something that might serve as a single unified front door for academia, for industry, and so forth. So we're just starting to think about that. I don't want to lay out any particular plans because they're sure to be wrong. But I talked about these amazing lab directors. I'm quite confident that they and their amazing teams, with input from you all, we can figure out what do you need from us, right? And how can we deliver that from the Department of Energy, because there is no other agency that's poised with all of this capability to do what it is we're here to talk about doing today. So, what can we create to power the bioeconomy at scale, right? The way we do things, the way we do in other areas. Confident we'll figure it out. I'm confident also that they will send me a bill. I'm sure it's not going to be cheap. We'll cross that bridge when we get there. That's fine, right? We already know that we're going to invest. We already have planted flags in this budget. Can't talk about next budgets, but the Department of Energy is moving out in biotechnology, in biosecurity, in support of, yes, the industry of the future, bioeconomy, but also all these other things, and not forgetting the national security piece. You'll hear more about this over time. As I said, we're kind of kicking off the planning process, kicking off the conversation about how that might work. But that one of the beauties of something like a mission agency like the Department of Energy, call it another superpower, if you will, we can move quickly. We can decide we're going to do something. We can pivot and marshal resources because we are a mission agency, and we do have these laboratories. So anyway, been fun to be here today. I don't think we're doing any questions, right? Am I over time? Okay, sorry about that. All right. Anyway, thanks a lot. It's been fun. I hope this is valuable for you all, and I do hope that you have the opportunity to provide the input for us. I'm sure we're going to find a way to do that. Looking forward to what we can do together. Thank you.
Wow, that was great. Okay, I'm standing in the way of what happens next. But before we do that, I want to make a few closing remarks. I was voluntold to make a few closing remarks. First, I want to make a call out to that amazing point that was made by Undersecretary DeBar earlier today, that a comprehensive biomanufacturing bill could really help drive the US bioeconomy. And I was listening today to all the great ideas and progress that's been made, and so right from the very start, that was pretty inspirational. We also heard about making DNA as a thing, driving the bioeconomy, making DNA, using DNA to program organisms and non-programmed organisms to make products and energy to drive the US bioeconomy. We also heard about how the food system is even more transformed by biomanufacturing than probably many of us realize. And we've also got a chance to do a little bit of a deep dive into how the convergence of artificial intelligence, automation, and engineering biology are changing almost every sector of the US bioeconomy. We were also reminded to be mindful of ethics, because as we live and work with these organisms that are self-replicating, with that comes responsibility. We also heard about the challenges of scale-up, policy, funding, flexible fermentation facilities, and we also heard about the challenges of workforce. Here at the end, Director Fall said, we need to be good stewards. We need to be stewards of good discovery science and translate that science to public benefit through innovation. 8 years ago, I was lucky enough to be the author of the National Bioeconomy Blueprint. And so thinking then about the vision of the bioeconomy and watching today what we've seen here, I could never have predicted artificial intelligence and automation and microfluidics and all of the things that are happening and how our students are being trained differently and thinking differently in multidisciplinary and interdisciplinary ways. With all of those things said, it's been an honor today to drive you through this emceeing activity with my colleague Blake Simmons, but we're not done yet. I hope you've made a lot of new connections, national labs and industry. I want you to join us upstairs, all the way up to the 8th floor. When it says transfer to the reception, it means walking out there, getting in an elevator, and going up 8th floor. There's some over there. Okay, go that way. And I think we have a little bit of a, first of all, the reception is generously sponsored by the University of California. And also, I understand that Impossible Foods may have put a little something on the menu for us. So here we go, biomanufactured food. With that, you're excused for now, and please go upstairs and come back and join us tomorrow for breakfast, and we get restarted again at 8:15 tomorrow morning. Thanks to everybody.